Good morning, everyone. I am here to introduce someone who needs no introduction. Our dynamite comes in small packages. Nokubonga. She completed her MSc here in 2015, 2016 through Fort Hare, but she was based at SAIAB, and we were lucky enough to get her back in 2023 for her postdoc. So her research focuses on water quality, marine pollution assessment, specifically contamination-related ecological and human health impacts. So her work deals mostly with those bioindicators that are edible, mussels, oysters, and limpets. And today she will be talking about ecological risk assessment of metal pollution in shellfish along the south coast of South Africa, implications for healthy and sustainable coastal ecosystems. Thank you.
Thank you so much, Sue, for such a great introduction. Starting from when I first got here to SAIAB as an MSc student. This is also very important for me, an important day, because the last time I gave a talk here, it was also during the World Ocean Day. And even this year, I'm giving a talk just prior that World Ocean Day, which will be on Sunday. And I just wanted to remind us or take us through what actually really is the Ocean World Day about. So this is a reminder to all of us to take care of our marine ecosystem, but also it is a call to action to all, and this is important for the sustainability of our ecosystem. Now, speaking of sustainability, what truly sustainability means, and I want us to examine in the context of seafood safety and also marine health. And I think that sustainability means that we do know that our coastal ecosystem are facing a challenge of pollution, and in this case, it includes metal pollution, which is what I'll be speaking about today. And the marine pollution, be it the chemicals or the microplastics we heard from the other talk from the other day, they threaten our biodiversity and eventually also threatened the seafood. Now, the marine pollution also does impact the fitness and the health of the marine organisms, and how they do this, they would impact their reproduction, and sometimes if these chemical pollution are available at higher concentration, they could also lead into mortality into some other species. Now, the assessment of the metal pollution along the south coast of South Africa, it is a critical aspect for sustainability. However, it is not well understood. It is really understudied. So the South African coastline, it is an important marine coastline, as it is also a hub to a diversity or a biodiversity of species. It is also important in the fisheries as well. And we do know that along the South African coastline, we have these ports to which there are different activities being done there, some ship repairs as well, all of which they do contributing to the contamination. So what our study focused on among these harbors, we focused on the port of Ngqura and the port of PE, among other sites that I will just show you as we move along. Besides the natural occurrence of the heavy metals or marine pollution, the anthropogenic influence, the marine pollution in the aquatic ecosystems. So this could be through the agricultural activities, that means the use of chemical containing fertilizers, or also through the industrial activities. And in this case, I just showed you a model of an industry depositing some contaminants into aquatic water system. So the obvious one, it would be the pipes, the discharge of these metals through the pipes, but also some river inflows, rivers containing metals. They do also end up, we all know that everything that happens in land, it all leads into our aquatic systems and all end up in the ocean eventually. So what I'm having here, it's a picture of a PE harbor. Even though we cannot assess the contamination, special chemicals through just visual appearance, but one can notice already that this water is not so clean, and we'll also see in the results, it will also reflect in the results as I move forward. So the filter feeders or the grazers, they are the organisms that are important in water quality assessment. They are regarded as bioindicators. But as much as they are bioindicators of some contaminants, they are also important in the ecosystems as they serve as a seafood, as you see the mussel dish there. And now the ability of these organisms to be able to filter or accumulate metals, they do really highlight a hidden risk to marine life, but not only to marine life, but as well as to human communities who really depend on these organisms for their sustenance and livelihood. And especially along the East Coast and the South Coast, we know, especially in the East part, we know local communities do rely on these shellfish species, your mussels in particular. Now, in this study, we assess metal accumulation in choosing sediments, seawater, algae, and invertebrates. We also wanted to examine the ecological risk posed by these pollution in the coastal ecosystem. But then I want you to know that the ecological risk assessment, we didn't do special for the purpose of this presentation. I'm not going to be showing some calculations, but what we did, we compared our measurement, the results that we have, to the standard regulatory limits of water quality. So we did set some expectations. So we said, because we're having urban, in urban environment, we have the large pots and the smaller pots. We said in urban areas will exhibit higher metal concentrations than the natural sites. And then the larger pots, we were expecting them to show higher metal concentrations in samples than in the smaller pots. The filter feeders being able to accumulate and or filter large amount of water, we're expecting them to accumulate more metal concentrations or elevated metal concentrations than the grazers. Also, sediment, seawater, and the diet being out, we're expecting those to be the source of metals. So we've selected these 6 sites. So the Port Alfred Marina, Cannon Rocks, Port Port of Ngqura, Port of PE, Kinney Bay, St. Francis Bay, two sites, Cannon Rocks and Kinney Bay being the natural sites, and the Port Alfred Marina and St. Francis Bay being the smaller ports, and Port of PE and Ngqura, as I've mentioned, being the larger ports. So we characterized those as sources, the sediment, seawater, and algae, and then we wanted to also understand, in order for us to understand the extent of metal pollution and how the invertebrates contribute in this process. We sampled filter figures, including mussels, oysters, and barnacles, and the different species of grazers, 4 different species of grazers. But for this presentation, I've just selected to present the highlights of the important results. So I won't be speaking of those different species of limpets into details. So this is just showing a process of what we went through in order for us to do the analysis of our samples. After we have oven dried our samples, we crush them and package them for analyzing these metals. So we've selected to analyze these 8 metals: your arsenic, aluminium, copper, cadmium, chromium, lead, nickel, and zinc. Now, because we don't have ICP-MS machine housed here at SAIAB, so we had some collaboration at the UJ, so we analyzed those samples in their analytical lab there. So what I want you to draw your attention into here, so the overall what we found between the comparing metals in sediments and seawater, we found that the sediments had higher concentrations of metals in general than the seawater. And St. Francis Bay, Port of St. Francis Bay sediments, they were accumulating higher concentrations of metals than in other sites. And I did notice, and I'm sure even in other studies, they have highlighted that in the Port of St. Francis Bay, there are also fisheries activities there. So I think beside the natural occurrence or geochemical origins of these sediments, maybe that might have also influenced those higher concentration, or the inflow from the nearby estuaries. Even though the Gamtoes estuary, I haven't heard of them having higher accumulations of metals, but these are interesting results to find, especially at the smaller ports than the larger ports. When it comes to water, the Port of Ngqura, it exhibited a higher concentration of metals in water than in other sites. But this was strange because it was not reflecting later on. You will see in the invertebrates tissue having higher concentration in water, but then not being reflective in the invertebrates, indicating that it's accumulated from water. But we'll see in the next slides. So the concentration of lead in water, it was also higher in the Port of Ngqura, and nickel as well. So when we compared our measurements with the standards water quality guidelines, we find that some metals like your arsenic, copper, nickel, and lead and zinc, they were within the standard regulatory limits. But at Port of Ngqura, in other side, but at Port of Ngqura, copper, nickel, and zinc measured in seawater, they exceeded these water quality guidelines, meaning the water in the Port of Ngqura are highly exposed to those contaminants. Then in sediment, copper was highly above the recommended guidelines. Here we used the Australian and the New Zealand guidelines as well as the Canadian guidelines. So when it comes to these standard regulatory guidelines, there aren't very much a lot of newly revised guidelines. Hence also, I've decided to check into other global assessments. So speaking of the concentration in invertebrates, that is our shell species. So noticeable, the pot of PE, it was exhibiting higher concentration of metals in the samples from pot of PE, show the higher concentrations of metal. And overall, the filter feeders, they accumulated higher metal concentration than the grazers, confirming our expectations. And the oysters were very favored the copper and the zinc as they accumulated these metals in higher concentration. But also at the pot of PE again, lead was very high in the penna species of mussel. And also what we also noticed is that in the natural sites, so it's seemingly the penna was accumulating higher concentration of the toxic metals, your cadmium, arsenic, as we also have noticed here at PE. Then when it comes to the cadmium concentration, so the grazer, species of grazer, that is your longicosta, Scutelastra longicosta, and penna, they were showing to accumulate that metal in higher concentration. And because this is a natural site, this is also concerning together with that one. This is also concerning, even though maybe the shell picking or yes, shell picking might not be that regular, but if the site is freely available for people to go and collect shellfish, then it is concerning to pose some health risk to the population that is exposed to these contaminants. Also, the barnacles were shown to accumulate high chromium, and then cobalt was available in higher concentration also in the species of limpets. So we also tried to show the effect or that could be caused by the three metals using the standard regulatory limits available. So we have used the lead and arsenic and cadmium, selected these because these metals are the ones that we were having newly, well, not so newly revised regulatory limits. So when we compared our measurements, we found that these three metals, arsenic, lead, and cadmium, they were above the regulatory limit when we speak of the consumption. That means they are not posing some health risk. So we also assessed whether the accumulations were through water or sediments, and then we found that indeed the accumulations were through water and sediments. And one would notice that now we're only having three metals. So we selected these three metals because they were occurring and they were not below the detection limits in all our functional groups that we've measured in. So you would also notice here at the natural site, which is Cannon Rocks, there were accumulations of metals either by filter feeders or grazers, both through water and through sediments. So there are different pathways where metals are being accumulated. And in this case, we do see that they were accumulated through water and sediments. In this presentation, I'm not gonna talk about the diet one, but also that's a general pathway that we know metals to be accumulated through. So there were some interesting cases where we found natural sites exhibiting almost similar levels of metal concentration, especially your aluminium, nickel, and chromium. Similar measurements to the those we measured at the port, especially Port Alfred and Port of Muruga. And the higher concentration of also metals in algae and grazers as well, they were indicating to be similar to those we found in the Port of Muruga and Port Alfred. Now, trying to make sense of this, because these are natural sites, this is not where the industrial activities is normally occurring. What could have been the cause? So it could be because of maybe the geochemical origin, or also it could be maybe the influence of the agallas current flow, hence a question mark. So we do know that the agallas current is flowing southwards along the east coast. So the assumption maybe it could be the flow of that current could be carrying some metal in it that would also be distributed in other areas, or maybe it could be the upwelling zone containing some contaminants in it. So this, I'm not sure of, hence the question mark. Maybe someone has done it, I haven't checked. So the metal pollution or marine pollution, it does result into poor water quality. And as such, this causes a degradation in the ecosystem. And this really does undermine the conservation investment. But more than anything, marine pollution does lead to the loss of biodiversity. And also, this could also lead into a decrease into the fisheries space. Just also going back to our sustainability. So sustainable fishing, it really means cleaner water, healthier shellfish, and a safer seafood. Now, there aren't many laws that are strict in terms of the regulating how the pollutions are being discharged. And I think it's important that the strict pollution discharge regulation and laws need to be enforced. And this is because the subsistence and the small-scale fisheries are vulnerable to these toxic seafood. And also, not only that's also the problem, the coastal pollution also does have an effect to the tourism, the food security at large, and the health of the ecosystem at large. So what our results really show, the species that we've used, these biogenic species, shellfish species, they have shown to be bioremediators of the metals. And these species are shown to be crucial bioindicators for restoring the urban coastal systems. And they also serve as a natural mechanism for water quality improvement in the urbanized coastline, and also in the natural environments. And I thank you.
Thanks, Max, for that really thorough and interesting presentation. Do we have any questions? Yes.
I can give you. Thanks for that presentation. I think it leads to important work recognizing metals in marine pollution. I want to ask a question about your natural sites.
Yes.
Like what else would you say there's contamination of metals there? And you gave an explanation of only sea-based or ocean-based. Could it be also that it's coming through from rivers and activities based in Philadelphia? It's might not have a port, but there's still activity there.
Okay. Thank you so much for your question. So the Port Alfred Marina and St. Francis Port, Port of St. Francis, they are small ports. So in this work, we regarded them as a small ports. Then the natural sites, we selected Cannon Rocks and Kinney Bay. So the metal portion that we found, higher metal portions that we found at Port Alfred, you are definitely right. It could be through the agricultural activities. It could be also through the, I think there's a Kawi River there. It could be the deposition from there. And also there's some boat cruise occurring there. And then we do know when it comes to boats and ship, there might be some accidentally spills of some chemicals in the boat or ship. So it's definitely true that, among other effects or impact.
Okay, question online. Thank you for a really interesting talk. Is there any chance of the pollution bioaccumulating up the food chain? From Josephine.
Yes, definitely. So I'm having another, it's not a presentation yet, it's a paper, and crossing fingers for it to be published so that I come here and talk again sometime. So they do bioaccumulate through food chain. So diet is one of the most noticeable pathway for the transfer of metals. And I think also other contaminants. Yes.
Any other questions? Shall we go with Matt first?
What are the common sources of arsenic that you could think of that might lead to those high levels?
So in industries, for instance, the use of maybe batteries or any paint containing the arsenic contaminants, then that could lead into those higher levels.
Yes.
This one, the Department of Water and the Environment does ongoing sampling in the rivers. Is there a similar organization that's doing ongoing sampling of water quality along the coast?
So along the coast, there used to be, I think it was a mussel watch count, if I'm not mistaken, yes. So that program used to do an ongoing, it was a program that was doing an assessment of metals along the coast using bioindicator species. But I do also know of CSIR. They used to also do an ongoing assessment of water quality. But then sometimes other monitoring programs, they don't use the bioindicator species. They will just do the assessment of contaminants in water and sediments. And I think some of the work that the CSIR has done is like that. They've done lots of work on water and sediments, but selecting specific sites. PE was one of them, and Port of Mocha, I believe so.
It seems to me that there should be ongoing monitoring.
Yeah, that we definitely need an ongoing monitoring. It is needed, but then I think because of funds sometimes, because even us here would want to go into further, maybe into other regions, but because of funds, maybe one will not be able to do that. So funding, I would say, is strictly restrict people in doing some other research more than anything. I think I saw a hand in Gwen.
Just a follow-on comment. If I'm not mistaken, SAN has recently gotten a CMP.
Yes, very excited.
So I would follow up this.
Any other questions?
Did you consider maybe moving around your sites, asking questionnaires on the sources of pollutions in the area and how they may have influenced your results?
So asking questions from
Or
like
just asking
community.
around the major sources of the pollution and how they may have influenced your results.
Or you mean incorporating with like the social aspect, like asking people?
Factories, just
Oh, like inquiring from
yeah.
factories. No, I didn't do that, but then knowing what factories produce and what they do, the assumption would be whatever metal containing products they use, if those things end up in the ocean or in the coastal waters, then they will be accumulated eventually by the organisms. So we didn't ask the industries. No, I thought you were asking about the community. Yeah. Anton?
Thanks for the opportunity. I was just wondering, like, in any of your sites that you measured, if there was a case whereby there was a direct correlation between the sediment concentration, water concentration, and also the uptake in the tissues, like whether all of those three correlated in any of the sites that you measured.
Yes, there were cases like that. For instance, cadmium at Kinney Bay, which is why I was saying it was an interesting case, because it's not big port, or it's not a place where there's lots of industrial activities around it. So there was cadmium. It did show that, and also arsenic, I think, and nickel as well. And then the other paper or the other data, it shows it nicely also how from the source to the bioaccumulator or to the species, how it's transferred. There were cases like that.
Did you check to maybe connect heavy metals or toxic metals with microplastics? Because that can also be like play a big role. Some toxic metals absorb to the microplastics, and that can be explanation for why the inverts show different levels and so on.
No, we didn't do that, and you and me, we shall talk. And so that's also another space also that I want to venture into, also the pharmaceutical space, also checking those, but yeah, we shall talk. We didn't include those. That would be amazing to see.
Anyone else? Yes.
Can I ask another question?
Tadiwa then.
I saw you mentioned something about the biodiversity.
Yeah.
Did you maybe already did another study how the pollutants affect the presence and absence of certain species, and also in sites that are polluted and natural sites, are there like differences in the morphology, like showing like whether these pollutants actually affect the species themselves?
And it's good that you speak of morphology. The PE in particular, where we find oyster, I think there's oyster farm as well. So the shell, you find out the shell is like bigger maybe, and then when you get inside that shell, you find out the tissue, the soft tissue that you thought or you assume it could contain a bigger tissue, and then it's tiny, or it's almost as if it's dead. And also the shell shape, they were different sizes. Some were very tiny in other areas where we were finding high contamination, but then we didn't directly assess or correlate these two. But that they asked that is that confirmed that the oyster content in the PE harbor, they are very highly affected by the water quality. But I haven't correlated these two. It's just a general notion that the higher the contaminants, and then potentially the production of the certain species, or it won't be as productive as one would expect. And also sometimes mortality could lead into other species, and also shift. Species will move into favorable conditions, I suppose, but I haven't linked those two.
Yes.
So I had a similar question to that. I wanted to ask if you recognize the morphology of a highly contaminated organism, which would be useful to pass on to people using that animal for food.
Yeah, and I think people would do also, sorry to cut, I think people would do also physiological studies. They might have also compared these two. I'm sure it is very much out there how the physiology or affect certain species, or certain metals affect certain species. Because in these metals, there are also essential metals like your copper, zinc, that are useful in the biological process of certain species. But then even though they are essential, once they are available in higher concentration, there are certain thresholds, then it becomes toxic to certain species.
Can I just follow another question? It's probably a very silly question, but does salinity affect metal bioaccumulation compared to freshwater? Is there a difference?
So for the purpose of the presentation, so I would have also included the physical chemical parameters affecting. So the physical chemical parameters, obviously, like salinity, it would affect the what is this? The dissolution or the speciation of the metals. So they do have some impact on the distribution of metals.
So they become more available or less available?
More available or less available, or the dissolved content of it become less available or more available, depending on the salinity levels.
We have two questions online. Tafara's links very nicely to that question. Great presentation. My question would be, did you measure any physicochemistry of the water? That could explain why you found high concentrations in water in one of your sites when there were low concentrations in mussels. For example, pH affects bioavailability of mussels.
Yes, so this question is exactly like the question that we're having here. So we did, am I speaking here with you? We did measure the physical chemistry of your water, your temperature, pH, and dissolved oxygen and salinity. But for the purpose of this presentation, so I didn't want to dive into those deep details or presenting the overall data, but they do have, there were some correlation where there's a lower pH, maybe you find a higher metal concentration, either cadmium or arsenic. But I can't remember from the top of my head which were those metals that positively correlated either to pH or salinity. But there were some measurements that we've taken, and they do have an impact.
Next one from online. Piwe says, thank you for the interesting and eye-opening talk. Are there any common sicknesses experienced by humans consuming these shellfish in the sites where heavy metals were above standard limit? Like, how do the high levels of these specific heavy metals affect human health?
AP, you are spoiling my other talk. Yes, they are. So yes, they are. So some metals like even the one that I didn't include here, or we didn't, in fact, we did measure, but it was below the detection limit, the mercury. Yes, mercury JP, I think. So there are metals that cause the respiratory infections, and also some could lead into cancer. They are carcinogenic. So there's a lot of range of things. The brain damages your kidney. So once they are above these regulatory limits, they do have some impact on human health, which is why we do need the fresh or newly developed standard regulation. So that when we compare these results, we don't use the old standard, we use the new standard limits.
Any other questions? All right, thank you all so much for attending, and thank you again.
Thanks, Yusuf.
Once we stop eating shellfish. It's something bad. Give them Canada.